Okay, since today we cannot meet and have lab and we have to do a virtual lab, the purpose of our lab today was going to be to examine the components of compounds. And we're looking specifically here at biological components. And I know we have not gotten to have the lecture material yet on organic compounds. Unfortunately, we're going to start that on Wednesday. So just very briefly, there are four primary types of organic compounds. One of these are carbohydrates. They're also called starches or sugars. These are hydrocarbons. It is mostly just carbon and hydrogen. These provide our bodies with energy. And we'll talk more about these on Wednesday. But today we are going to perform a test for starches. Our test for starches will involve taking a solution, which we want to examine. You place it in a test tube and you add iodine. Iodine, when it is not exposed to starch, is a brownish color. When exposed to starch, iodine turns to a purple. So we'll be testing starch by using iodine. Another organic compound we're going to look for in our solutions are lipids. Lipids are also called fats. This is present in vegetable oils. It is present naturally occurring in other plants, such as the avocado. Lipids can also be found in animal products. Think about a steak. So that white, fine grain that makes it so delicious, that is a fat. That is lipids. So we're going to also test for lipids. The way we test for lipids is by using a solution called burette solution. Now, in this virtual lab, there is no burette solution. We do have that in our real lab. So it is actually a combination of sodium hydroxide and copper sulfate. Even though this virtual lab does not provide us with just a pre-mixed burette solution, it does provide us with copper sulfate and sodium hydroxide. And we will be combining copper sulfate and sodium hydroxide to make burette solution in this lab. The final test All right, I know we were all looking forward to doing a bit of chemistry in lab this week, but since I cannot be here, we will have a virtual lab. Now, this virtual lab is going to be very similar to what we've done in class, just a little less fun because you don't get to play the chemicals yourself, and I apologize for that. However, what we will be doing in lab this week is testing for biological compounds. These are compounds we will discuss more on Wednesday, but I'm going to go over them very, very briefly with you before you start this lab. There are four primary types of organic or biological compounds. Those are carbohydrates, proteins, lipids, and nucleic acids. We will not be testing for nucleic acids in this lab. We will be testing for starch, for lipids, and for carbohydrates, for lipids, and for proteins. Now, starch is a type of carbohydrate, and we will use iodine to test for our carbohydrates or starch. The way we'll do this is when you add iodine to a solution that contains starch, iodine changes from brown to purple, and this will indicate for us that a starch is present. Starches are also called a large type of sugar or complex sugar. And so we'll talk more about these compounds in lecture on Wednesday, but for right now, we're just going to work on testing for them. Another organic compound we're going to test for are the proteins. Now, the way we'll test for proteins is by using a solution called burette solution. Now, in lab, I have pre-mixed burette solution, which is very simple to add. However, in this virtual lab, they have not provided us with pre-mixed burette solution. They have provided us with copper sulfate and sodium hydroxide, which conveniently is exactly what burette solution is made out of. So what we do is we combine these two, the sodium hydroxide and the copper sulfate, with the solution we want to test for protein. If the test is negative, it will be blue. the copper sulfate is already blue, it remains blue. If the test is positive, it will turn sort of a bluish purple, much like the iodine test does for starch. Now, the last test we'll conduct in lab will be for lipids, which are also called fats. Now, this test in the virtual lab is the one I am least satisfied with by far. I think the test results are not as clear as they would be in lab. I don't really like how it's presented, but we're going to make do with it. So when you add Sudan-3, which is the solution we use to test for lipids, it will go from a brown color to a less transparent or harder to see through, sort of reddish color towards pink. We would not use Sudan-3 in lab. We use what's called the paper bag test in our lab. You take a brown paper bag, you lay your test item on the bag. It will leave a wet spot. You pick it up, and if the wet spot disappears in about 15 minutes, that was most likely due to water. And if it is still present, it is because it is from a lipid, which if you've ever seen an oil stain on concrete, that will not dry out and evaporate. So what we are going to do is I'm going to show you how to do this lab. I'm not going to walk you through the entire thing, but you will be testing for each of these different organic compounds and a few different solutions. And I just am going to provide you with a little more information when you read the rest of your lab document. Okay, so to do this lab, you click Begin Lab, pick your set of gloves. I happen to like blue. It will take it just a minute to load, and then you'll be able to start your lab. I'd recommend you zoom out a little bit because what you'll have to do is pan back and forth using these gray arrows to move from left to right. You can also tilt the screen up or down, which I don't think is really very necessary once you get it centered. Now, if you will look at this first set of test tubes, you will see we are going to set up our controls. Now, if you'll remember, the controls are what we use as our baseline data. We're going to have a starch control, which will be just water, then we'll test for starch. We'll have a protein control, then we'll test for protein. Over here we'll have a lipid control, which again will be water, and then we'll test for our lipid. I am not going to walk you through this entire lab, but I want you to see too, you have pipettes here. One is labeled water, one is labeled milk, one is labeled liquid. Your lipid is your vegetable oil, so when you need to use a lipid, pick up this dropper, put it over the vegetable oil, click it once, and then we're testing for lipids here, so I'm going to put it here in the test for lipid. Then you simply put up that pipette. The one for water works the same way. We have 3 test tubes that are currently calling for water. You add water to all 3, you put that back. Oh, and I grab the milk. Do not grab the milk, grab the one that's labeled water, please. And then when you need to add milk, you're going to use milk to test for the protein. And then for the starch test, you will use the container labeled starch. Now, to test for starch, and this is about as far as I'm going to take you, so pay attention. You add iodine to your test for starch, it turns purple. You add it to your control for starch, which water will not have any starch in it. This is just distilled H2O. This is your negative test. So in a little while, you're going to compare other solutions to this test. If it tests negative for starch, it will be this brown color. If it tests positive for starch, it will be purple. Now we're going to do a protein test. You can see a positive and negative result for protein. The protein is the one I told you. We don't have the burette solution. We'll have to use a combination of sodium hydroxide and copper sulfate. You add these two to your water. This is your control. It is just plain blue. There is no protein in the water. You take your sodium hydroxide, add it to your milk. You add copper sulfate, and it turns out grayish purple. This is a positive test result for protein. Now the last we're going to do is our lipid. This is the one I told you I do not like. Add Sudan-3 to your water for the control. Add Sudan-3 to your lipid, which is your vegetable oil. What I do on these is I will stir these up using my stirring rods. Stir your test for the control. get a fresh stirring rod, stir the lipid. If you will look at these, there is a color difference. If, for the purposes of this lab, if it looks different from your control, it is positive for lipids. I'm going to take you over here real quick. I want to look at these closer. So when you add these different test solutions to your unknowns and your albumin, what you will look for, a positive test for starch will be purple, not brown from the iodine. A positive test for protein is a sort of grayish color, not blue. Please remember you have to add your copper sulfate and your sodium hydroxide in order to have a burette solution. Now, what you will do in addition to this, you will take your albumin. You will test for albumin. You do not have a set of test tubes for albumin. You can use the unknowns. You will add albumin to each of these, test them for starch, proteins, and lipids. Test your milk for starch, proteins, and lipids. Once you are done doing that, if you have added, come here, if you add albumin to these, once you're done, you can pick up these test tubes, take them over to the sink, and dump them out. Once you have dumped these out from your test for the albumin, you will test your unknown. You will record your results, and you will also need to take a screen test for me that shows you have worked on this lab. If you have never taken a screenshot before, what you do is look on your keyboard for a little button that says PRNTSC or SCRN. That is print screen. You press that button, it will take a picture of the entire screen. You go into a Word document, you hit paste, which is on your mouse. You right click, go down to paste, and you click on that. It will put the image into a Word document. If you are comfortable with that, that is fine. If you know how to use the snipping tool, and I will put links to this information in your assignment, you will use the snipping tool. Looks like a little pair of scissors. If you do not know where this is, you can go down to your Windows and search for this tool. Just put in SNIPPING. The tool will open up, gives you a pair of scissors. You select the area you want to copy. You can't see anything that I'm doing. Let me make this window of what you're seeing larger. I don't know if I can resize it now. Okay. It will say you can save your clip. Click save, save it, and then you can paste that or add that into a Word document. I know that was very confusing. There is a better version on the website for the class if you go to this lab that will show you how to snip and copy and paste. If you cannot figure this out on your own, do this lab in the Academic Success Center and ask for help there in taking a snip or taking a screenshot and placing that in a Word document. Okay, what you will have to do with this data is type it up in Word and submit it to the Dropbox. Please notice that your lab has some specific questions for you to answer. I want you to answer those. Send me your data. You'll have a hypothesis you have to submit regarding your albumin, your milk, and your unknown. If you have any questions, please email me, and I will be trying to keep an eye on my email as much as I possibly can. There is a second part to lab where I will ask you to draw something and bring it with you to class for your next lab. So please also watch that video. Thanks so much. I apologize for the fact that you're doing a virtual lab this week, but at least a virtual lab is better than no lab at all. Like I said, please email with questions, and I hope you have a great day.